Good morning, everyone. We do apologize for the late start. It is indeed a pleasure to have you joining us today for another one of our webinars as we celebrate Agriculture Month under the theme, Transforming Our Food Security, Achieving Food and Nutrition Security. If you've had the pleasure to join us on Wednesday, you would have learned about rice cultivation in the hinterland regions. Today, however, we will be focusing on a very important aspect of rice cultivation, and that is disease management. You'll be learning about the major rice diseases in Guyana, their causes, symptoms, and also management practices that can be utilized in controlling these diseases. Before we get into our presentation, we'll have a few remarks from our General Manager, Mr. Kuldip Ratnat.
I extend to you on behalf of the Guyana Rice Development Board a pleasant welcome to this webinar on the management of rice diseases in Guyana. Rice diseases have been present in the industry since the 1900s. It continues to be important in limiting profitable rice production in Guyana and other parts of the world, and is considered to be one of the most in Guyana. Rice diseases can result in losses of up to 100% crop if not recognized early or at the right time, and adopt the appropriate management strategies. Today's webinar is aimed at all our viewers, wherever you are. I extend to you on behalf of the Guyana Rice Development Board a pleasant welcome to this webinar on the management of rice diseases in Guyana. Rice diseases have been present in the industry since the 1900s. It continues to be important in limiting profitable rice production in Guyana and other parts of the world, and is considered to be one of the most important factors that affect rice cultivation and its productivity in Guyana. Rice diseases can result in losses of up to 100% in a crop if not recognized early or at the right time, and adopt the appropriate management strategies. Today's webinar is aimed at discussing the major rice diseases in Guyana, how to recognize or identify them based on symptoms under field conditions, and how they can best manage using the recommended integrated disease management strategies that has been tested and developed through extensive research work carried out by the Plant Pathology Department of the GRDB. The board continues to support research in this discipline and others as well, in order to reduce the losses that are incurred by farmers, thereby enhancing the performance of their crop. I invite you to sit back, pay close attention to the presentation to be delivered by Dr. Rajendra Prasad, our plant pathologist and head of department. I thank you.
Now for the main item on our agenda. Today, we have with us Dr. Rajendra Prasad. Dr. Prasad is the plant pathologist at the Rice Research Station. He has been with the Rice Board for over 2 decades and serves not just as the plant pathologist, but as the HOD of the Plant Pathology Department. One of his main roles as the plant pathologist is to determine effective ways of managing rice diseases. Today, he will be presenting to you on management strategies of major rice diseases in Guyana. Over to Dr. Mahendra.
Good morning, everyone, and thank you for joining us. It's indeed a great pleasure to give this talk during our Agriculture Month as part of the GRDB activity. So my role is just to go through what are the major diseases affecting the rice crop and how they can be effectively managed. So basically, the presentation would follow where I'll give you a brief overview of the of rice, and then look at the major diseases, some of the causal organism, the economic importance or losses that these diseases are capable of causing, the major symptoms, which is one of the most important things we stressed on, is the symptoms, how to identify these diseases under field condition. And also we'll highlight some of the condition that favors the disease development. Then we look at the management strategies. Rice, as we all know, is one of the most important crop plant on earth. And it's a major staple food on more than 1/3 of the world population. In Guyana, it is one of the most, in fact, it contributes more than 20% of the agriculture GDP, and it's considered one of the most important agriculture industry. Apart from being the largest agriculture users, it also provides support directly and indirectly to more than 10% of the Guyanese population. Now, we can't overlabel or talk much about the importance of rice, because it's much to be discussed. However, in Guyana, rice is primarily grown in region 2, 3, 4, 5, and 6, and only recently in region 9, the hinterland area. And in these different geographic area, the rice crop suffer losses from major disease. Luckily in Guyana, from all our survey as recent as last crop in 2021, the 5 major rice diseases that we have observed throughout the rice industry start off with the rice blast, brown spot, sheath blight, sheath rot, and the grain discoloration. Now let us look at the rice blast. Rice blast, and all, before I move on into the blast, we are lucky to have all these disease to be caused only by fungal origin. So they are more easily, we are able to develop strategies that we are able to combat these very in an easy way, compared if you have to deal with other strains or category such as virus, bacteria, and so on. So we are lucky to have only these major diseases of fungal origin. So in terms of the rice blast, it is caused by Megnaportia oryzae, which is a sexual form, and Pyracularia oryzae, which is a asexual. And this disease is capable of causing more than from 5% to 100% losses in terms of crop yield. And if it is left unattended and not managed properly. And this disease is capable of affecting all parts of the plant and throughout the growth stage of the crop. As you can see on your screen, that's how the spores basically of the blast Pyracleria is basically pear-shaped, and it has two septa, one and two septa. That's how we basically identify the pathogen under laboratory condition using a microscope. Some of the condition that favors the development of this disease is the presence of the pathogen or the blast spore. We have low soil moisture, prolonged rain period, or we have an intermittent period of wet sunshine, rain, dew, these kind of condition, cool daytime temperature and nighttime temperature as well. These are all conditions that favors the development of blasts, and this can even get into epidemic proportion. Now, based on the part of the plant that is being affected by the disease again, the name of the common the symptoms. For example, leaf blast basically first appear as a gray-green water-soaked lesion, which has a dark brown border towards around the pathogen, the lesions. These lesions are the initial lesion, and as they become advanced, the lesion form typically into an elliptical spindle shape with an ashy gray center with a dark brown margin surrounded with yellow hollows. And all these images on the side of the screen, as you can see, shows the initial symptoms, and as the symptoms develop and into the more typical blast symptoms, the leaf blast. As we mentioned earlier, the blast pathogen can also affect the color of the plant, which is the neck, or the color, and it forms a necrosis at that area, joining those tissue. Likewise, on the neck of the panicle, and on the panicle itself, it forms lesion-like appearance on the panicle branches. And when the infection occur at an early stage, it causes the grains to not full, to fall off, to shatters, and so on, and you cause a reduction in terms of the number of grain, the quality of the grain, and so on. This disease also affects the seeds and form dark brown lesion appearance on the grain itself. Now let us look at the other major disease is the brown spot. Brown spot basically caused by Polaris oryzae, and is capable of causing yield loss and with moderate incident from 12%, and in severe case up to 45%. However, there are cases in India, state named Bengal, if we know our history, in the earlys, it caused 50 to 100% yield loss, because the condition during that period was favorable. You had cool nighttime temperature, high relative humidity, and so on. And the condition was so favorable, and the persons were more depending on rice, as I said, as a staple food. So you had a death toll in greater than 2 million persons. So these diseases are very serious when the condition is correct and they meet the epidemic proportion. Right. So like the blast, the brown spot can also affect the crop at all stages. And as I mentioned earlier, some of the condition, high relative humidity, temperature between 16 and 30 degrees. And one also is unflooded or water stress condition, also predispose or make the condition more favorable for brown spot attack, as well as nutritional deficiency. As we know from all our soil test analysis done, from throughout all the region, we have found that some of the soil are deficient in elements such as P and K. And more so, most of the soil are deficient in the K. And as we know, the nitrogen, if it apply at a high concentration or if it's overdosed, you would make the plant more vulnerable to the disease. So in this case, we are already seeing some of the strategies that we can utilize in terms of management as we move along, even though we didn't reach to the management aspect as yet. So nutritional deficiency is a key factor in terms if you want to manage your brown spot incident as well. And some of the other major sources of infection or that can predispose the crop to a higher level of brown spot is where you have when you have used infected seeds, or you have volunteer plants, red rice, and other infective debris, weeds such as board seed grass, and so on. These are all alternate hosts that the disease can survive on throughout the season. Now, in terms of looking at identifying the disease under field condition based on symptoms, as we mentioned, it can affect the crop from all stages, from seedling to maturity. So at the seedling stage, it appears as a small circular yellow-brown lesion on the coleoptile of the rice plant, as you can see in the image at the side. Likewise, in the tillering stage, the initial lesion are small, circular, dark brown to purple. Again, you can see that in the picture at the side. And as the lesion develop, they become oval to elliptical shape. Some are irregular in shape. They have the same ashy gray center with a dark brown margin along with a yellow hollow surrounding the lesions. So sometime or most of the time, farmers interchange or confuse when they're looking at the symptoms of the brown spot and the blast. So one of the key differentiating factor is looking at the lesion, the shape of the lesion. As I mentioned, the blast lesion is diamond shape or spindle shape, while the brown spot is irregular, oval to elliptical, right? An unsusceptible variety, these lesions come together, coalesce, and form larger, cover larger surface area, and can cause the entire leaf to wilt and the plant to die. While in resistant variety, when the variety resistant kicks in, what you would only find is tiny brown specks on the leaf. And you can see in the images showing the comparative view. So what happens there is the resistant mechanism kicks in and create a hypersensitive lesion. And the lesion would not further develop into a typical brown spot lesion. Also, you'll find the grains being discolored when they are infected with the brown spot symptoms. Now let us turn our attention to the sheath blight. Sheath blight caused by Rhizoctonia solani, and it is capable of causing a yield loss 20 to 50% on susceptible cultivar, and it can affect the plant from seedling stage or to tillering stage as well. And the images on screen shows what the mycelium look like. And if you look carefully, you would see these are a typical Rhizoctonia mycelium. You would see they have a right angle branching from the mycelium, and you have those septa forming in between. Those are the key features when you look to identify this pathogen under the laboratory condition. And when these mycelium get old, maybe 7 to 10 days, they form what we term as sclerotia, which is the resting stage of the fungus. And this basically happens also under the field condition. And this resting stage is the pathogen is able to survive for more than 15 to 20 years using this sclerotia. So, and they can move, this sclerotia can float in the waterway and move from one field to the next. So as we go along, we're discussing, and then you will see at the end how we can manage the disease. Some of the condition, again, favorable is likewise high temperature within 28 to 32 degrees, high level of nitrogen, and high relative humidity above 85%. What the nitrogen does, as I mentioned, it makes the plant more succulent and easier for the pathogen to penetrate if it's above the recommended rates. Likewise, if you use a high seed rate, it could create that micro environmental condition within the plant, making the plant more susceptible or vulnerable to develop the disease infection. Now, the typical sheath blight symptoms usually occur above the waterline under field condition. It's basically an oval or ellipsoidal grayish green lesion, and these lesions can range between a 1 to 3, and even in highly susceptible cultivar, these lesions coalesces together and even can take the entire tiller or plant. Now, likewise, sheath blight also affects the leaf, and it forms irregular shaped lesions with gray to white center and brown margin. But under our condition, we are finding more so not so much of the leaf infection, but the tiller and the entire plant. In terms of sheath rot, this pathogen is caused by Sarclodium oryzae. It is one of the most destructive pathogen to emerging panicles. And if it occurs at an early stage, losses can range from 20 to 85%. And as you can see, that's how the mycelium look under the microscope. Some of the condition favorable for sheath rot development is they are more prevalent, like now when we have the wet condition, increased plant density or high seed rate, likewise high nitrogen and high temperature and relative humidity above 85. So most of the diseases favor, or they're favorable when you have a certain degree of temperature as well as relative humidity, mostly above 85, and if you have high nitrogen, high plant density, and so on. All right. So basically, the symptoms as it appears, it starts on the uppermost leaf sheath of the rice plant. They are oblong or irregular, spot with dark to reddish brown margin and gray to green center. And they can, as I mentioned, it can affect the entire panicle. And below you'll see some of the images where they cause entire leaf sheath to become discolored, brown discolored, as well as the grains, and the grains also fail to develop. All right. Here you see the panicle fail to emerge because the disease occur at an early stage, and all the grains within that panicle destroyed. Right. In terms of grain discoloration now, grain discoloration can be caused by an association of many microorganisms such as Alternaria, Calvularia, Fusarium, Dretzschlaria, Oryzae, Pericularia, Oryzae, Circladium, Oryzae, Sclerotium, Aspergillus, Penicillium, and so on. And as we know, over about 2 years ago, pharma has discovered this grain discoloration as being a problem where it's affecting their grades at the mill and so on. And there was some confusion with the millers are terminated as dirty grain and black tip. And they were penalizing the farmers as damaged grain. And we have embarked on a research in order to find what was the cause and how to manage, how we can better manage the condition. But before I go to that, let's look at some of the symptoms. So the symptoms basically appear as a dark imperfection on the dorsal surface of the grain and has a lesion-like appearance. Now, when you shell the grain and you see that discoloration, and you can use your tip of your finger and if you rub it slightly or scrape it, it would fall off, right? So, and this condition, generally, you cannot detect it before harvesting because it's inside the grain after you shell the grain. So what we have found from our studies, we found Calvularia lunata to be the most prominent pathogen on the grains that had the symptoms of discoloration. We also found Bipolaris oryzae, Celcrodium, Alternaria, and Aspergillus, as well as Fusarium, at very low level on the said grains. And this, we seek some help from CABI, and they did a detailed analysis, and they have found Calvularia lunata to be the prominent organism up to a DNA level. They have undertake that piece of study. So in terms of management strategies for these major diseases, the first and foremost is the use of resistant variety is our number one priority. And over the years, the GRDB has embarked on a program to evaluate on a seasonal basis advanced germplasm material emanating from the plant breeding department, where they acquire through their local crosses or hybrid program, as well as from accession received from other international bodies such as the FLAR, CIAT, CABI, and so on. These lines are tested in hotspot location across the country, and those that are found to be resistant are further selected and developed by the breeder in order to be released later as variety once they are found to be suitable. All other factors being equal, mean plant type, grain type, and so on. So one of the selecting factor is blast resistant, and that has been the main role or main focus of our disease management program, since blast, as we know, is one of the most destructive pathogen in more than 85 countries of the world that grows rice. So and this pathogen is so difficult to manage because it changes its form, it changes its strain, it mutates. So one variety can be released in, let's say, 2021 as being resistant to blast, and immediately after it can be hit by the blast pathogen, meaning the pathogen can change its form a new race or new strain and overcome that resistance in that plant. So that's why it is paramount important for us to continue to search for new option or rice variety that or rice lines that has resistance in them, right? Apart from that, we are also, our program gears look at variety that has tolerance to brown spot, sheath blight, and sheath rot. We have not ventured out to look at for tolerance for grain discoloration at this junction as yet, but it is a plan for the upcoming research. So also, all these variety that has been released have shown high level of tolerance to the other three major diseases I ever mentioned, right? So in terms of blast resistance, since blast is one of our major concern, where we are with that. So far, we have more than 17 variety released by GRDB, and we have found up to 2020. Some of these variety still maintain, or most of them, the GRDB 9, 10, 12, 14, 15, and 16. I didn't add the 16 here as yet. This 16, all these variety releases resistant and still remain or holds the status of resistant. While we have some of the older variety that were released as resistant, for example, the G98, 196, the 135, right, and so on, they have changed from being resistant to highly susceptible. So that to show and that the blast pathogen keep changing and can overcome the resistance in the plant. In terms of cultural practices for disease management, it is recommended to sow within the recommended cropping period, and that we know. Practice proper field sanitation, as I mentioned earlier in the presentation. The pathogen can survive for prolonged period on alternate hosts, plant debris. So it's important to practice proper field sanitation. Make sure you clean around your field, burn all the debris and so on properly. Do not have excessive plant density. You're looking for an ideal plant density within 100 and let's say 125 to 215 plants per meter square. That would, when you use probably 100 to 140 pounds of seed per acre, you would almost get the ideal plant density which would also help in terms of your disease management approach. Avoid application of excessive nitrogen and also keep the soil nutrition status in this balance. So that can be done through using split nitrogen application, as well as look at your nutrient status, see if your soil is deficient in any of the P, K, and other elements, and do your soil amendment. As I mentioned, any stressful condition predisposes the crop for disease. So advise to keep your field well irrigated, well flooded. Get rid of alternate hosts, as I mentioned. Also, from our earlier research work, we have found some very cheap and environmentally friendly approach in terms as alternative in terms of managing disease. Use of black stage, bale, and madar. You can find this anywhere you drive around the country or in the back dams. These are plants that have more so of weed of origin in Guyana, and these plants has found to have high antifungal properties against the blast disease, and show superior control. Likewise, we have found the lemongrass and the thick leaf thyme to be very effective against the sheath blight disease. So these are all options available to you. In terms of the use of bioagent or biocontrol agent, we have the Bacillus cereus OG2L and Bacillus subtilis OG2A, are effective against the blast, and we have the OG2L effective against the sheath blight. Now, these are recent finding, and we are presently the body is working to make them available in terms of more increasing their shelf life and make them into proper package product that can be sold to farmers in Guyana for use. Now, in terms of the use of recommended fungicide and use them at the corrective rate, this is the last option. And in most cases, once you use the other management option, there's limited need for the application of fungicide. However, we are asking that you don't apply fungicide as your force mean defense mechanism in the event of or in your approach of managing disease, right? So some of the recommended fungicide and effective against is the Anthracal, the Nativo. The Anthracal is effective against all the 5 major diseases. Likewise, the Glory, the Amistar Extra, Carbendazim, Fuji-1, Superblast, and Manzate. And you have the rates here as in given. Now, these products are recommended to be applied in 2 motor blowers of spray solution per acre in order to have the best and more effective control using the fungicide against these particular pathogen. From the research as well, and if you so desire, all these findings that we have presented here today are published in the international journals, and they are available online. If you want further reading, you can go to them for more detail and specific. But what we have found, when we have used these bioagent, chemical, and plant products and so on, we have seen a positive influence in terms of the rice crop growth as well as the yield. Now, what can happen if you fail what can happen if you fail to manage your disease or you neglect the disease? And at 5% losses, and we use an average of 40 bags per acre, you're losing 2 bags per acre, which is approximately at a rate at let's say a bag of paddy is about $3,000 per bag. So it's about approximately 6,000 when you only have 5%. And as the percentage losses increases from 12, you have 14,400, then you have 24,000. So as the incident of disease increasing in your crop and the losses increase due to the disease and due to the neglect of not taking the management approach on a timely manner, you are losing money. Because we have estimated from these recommended products, we have estimated that for treating 1 acre, it would cost you minimum $1,000 to $3,000. So we're advocating rice is a business. Do it as a business. Manage your disease. Minimize your crop loss. maximize your profit. Before further ado, I must give special thanks to Chairman, Board of Director, Management, staff of GRDB, and my staff, Ms. McGowan, who introduced me. She's one of our lead scientists with the plant pathology department. I must thank her and all the team on board for support and looking forward for continued support on our journey. Thank you, and back to Ms. McGowan.
Thank you, Dr. Prasad, for that interesting and informative presentation. Having learned of the importance of rice production in Guyana and the major impact diseases can have on that production, I trust that our viewers were able to appreciate the work being done to develop strategies in order to manage these diseases. We'll now open the floor for any questions that you may have. Please feel free to ask. If there are no questions, I'd like to thank you all for joining us. Please be reminded that our presentation will be available on the GRDB's website, Facebook page, and the YouTube channel. This brings us to the end of our webinar presentations for Agriculture Month 2021. Please feel free to join us again next year. Thank you.